So next our topic is biosynthesis of urea. What is urea, you know, right? Our body, after metabolizing carbohydrates, fats, vitamins, everything, they will form what? Ammonia. This ammonia will be detoxified in our liver. So they will produce what? Less toxic urea. So it will be excreted out, right? Now we will see how the urea is formed. Okay, there are some steps, and we have a great cycle, that is our urea cycle or ornithine cycle. Now we will see what are the steps taking place here. First one is transamination. Second one is deamination. Deamination, particularly oxidative deamination. This 1+2 step you can call it as trans-deamination. And third step is transport of ammonia. Transport of ammonia means from muscles that ammonia is transported by what? The amino acid alanine. From brain and heart it is transported by our glutamine. The last step is disposal of ammonia. Okay. Now we will see each step correctly, how each step works. Okay. So first step is our transamination. So what is this transamination? Okay. Now before that, see, just this is a tissue. This is a heart tissue. What is happening is, I am talking about all process, not about transamination. So what will happen? So the amino acid in the tissue will be transaminated to form glutamine. Okay. This glutamate will reach where? The glutamate will be reaching in our mother, that is our liver. Okay. This transamination is tissue level. Here we have one amino acid, right? The organ or tissue. So glutamate, the transaminated to form glutamate. So glutamate will deposit what? Ammonia here. So glutamate will be here. This process is known as what? Deamination. Okay. So glutamate will be here. Glutamate will come here and release ammonia. It is known as deamination. It is tissue level. This is our mitochondrial level. Clear? So these are the things happening. Now we will see each steps. So first step what we have studied is what? Transamination. So what is transamination? Transamination is the process of transfer of amino group from transfer of one amino acid to other. So what is amino acid that we have seen here? That glutamate. So that is transfer of, just write that definition, transfer of amino group from one amino acid to other. What is the other amino acid here? Mainly our glutamate. Okay. Now you will ask me why the glutamate is chosen here, why other amino acid? Because glutamate is easy to deaminate. So easy deamination is possible for. Deamination means what? Just dumping the ammonia to the liver. Easily deamination is possible for our glutamate. Okay. So we have studied this tissue level, and no ammonia is liberated, right? No ammonia is liberated. Now, what are the enzymes taking place in this one? That is transamination, right? Amino transferase. The enzyme is amino transferase, or you can call it as transaminase. This is a reversible enzyme. No, the steps can go both forward and backward. So reversible means you know it, right? Now the coenzyme here is also what? Coenzyme is vitamin B6, the PLP, pyridoxal phosphate. Okay. Now we'll study two important transamination. I'm just giving you examples of transamination. First one is alanine. Alanine plus alpha ketoglutarate acid. Okay, alanine plus alpha ketoglutarate acid, they will undergo transamination. Transamination, what will be the enzyme? Alanine transferase, right? Or transaminase. The enzyme will be transaminase. Now, and they will have vitamin B6 cofactor and transaminase enzyme. So they will form what? Alanine will form pyruvate plus glutamate. Pyruvate plus glutamate. Now, one more example I am giving you, aspartate. Here the keto acid alpha ketoglutarate is same. Okay, same, it is also undergoing transamination. The vitamin B6 and the enzyme will be aspartate transaminase, right? Aspartate transaminate. You can write the enzyme name here, SGPT. Here it is SGOT. Aspartate, they will form oxaloacetate plus glutamate. These are reversible. I have given you this reversible. SGPT means serum glutamate pyruvate transaminase. Very easy. Serum glutamate pyruvate transaminase. Here SGOT means serum glutamate oxaloacetate and transaminase. Clear? Very easy to remember, right? Now, what is the clinical importance of this enzyme? Now, if you these are asked in many MCQ. So, if you want to remember, I will give you a shortcut. Just remember salt. Okay? Just remember salt. You know soap, right? Just make a division. When it is aspartate, then product will be what? Oxaloacetate. When it is alanine, the product will be pyruvate. A is common here. It is alanine, product here is pyruvate. If it is aspartate plus ketoglutaric acid, the product is oxaloacetate. You can remember like this. Now, what is the importance of this SGOT and SGPT? Now, there are your neighbor or someone or in the hospital, you will get liver function test, right? So you can see in that normal SGPT and normal SGOT, right? They will give you normal SGPT and normal SGOT. If SGOT is increased, the liver failure is due to alcoholic reason. Okay? If SGPT is reduced, it is non-alcoholic reason. How I remember is, you can see in SGOT we have what? O. In vodka also we have O. So if people who are drinking lot of vodka will have more SGOT. So other in SGPT. So that you can remember. So in biochemical test, if you get a test, just look for the enzyme. If the SGOT is high, you can tell Dadaji, so drink less. Or if SGPT is high, it means what? Non-alcoholic fats or junk food. Okay. So if the SGOT level is high, it means the patient is too alcoholic. That's why he is having liver failure. Clear? Now, next topic what we are going to discuss is, not next topic, we have done with the transamination. See, transamination, no, all we Indians know IPL, right? IPL. Just make it as TPL. So there are three amino acids which doesn't undergo transformation. T for threonine, no tryptophan and tyrosine, remember that. P for proline and L for lysine. Okay, so these three amino acids doesn't undergo transamination. Here, these three amino acids doesn't undergo transamination. Now, second step what we are going to study is what we have studied is deamination. That is our oxidative deamination. Right? So next step is oxidative deamination. So from the word only, we are depositing ammonia to the liver. You can write the definition here: removal of amino group, that is removal of amino group from the amino acid, right? Removal of amino group to the liver. Now the steps, these are occurring in the mitochondrial level, right? It's occurring in mitochondrial level. Clear? Everyone is clear? Clear, right? Now, glutamate plus step is glutamate plus water. So here the equation, the process is deamination, right? So the enzyme here will be glutamate deaminase, right? No. So glutamates plus water, glutamate dehydrogenase. So what they will form? Then we are removing the ammonia, right? So here we are removing ammonia. So here the one product will be already alpha ketoglutarate is there, then NH4 plus. So the coenzyme here is NAD. NAD is for our nicotinic acid, that is vitamin B3. Okay. So this is the second step. Now third step is what? Step 3. Step 3 is what? Our transport of ammonia. We have already studied from muscle who is transporting alanine, and from heart and brain is glutamine. Clear? So this is the third step. Now the fourth step. Fourth step is disposal of ammonia. Disposal of ammonia, this is our important cycle. What is that? Our urea cycle or ornithine cycle. Urea cycle, or you can call it as ornithine cycle. One more name is there, that is a special name.